So Andrew, tell me about how did you arrive at this topic, or what other topics were you looking at? What was your background in deep learning?
Yeah, so I got started with like deep learning probably like my sophomore year. Kind of just some like introductory projects, kind of self-studying it mostly. And I really got interested in it after like looking up some of the applications of deep learning, and especially like because I didn't really know how drugs were made. And it was kind of like a Google search that I did like one day randomly. And I found out that like a lot of drugs, they're kind of just represented as these compounds that can be um, interacted with using natural language processing and neural networks. And yeah, that was kind of my intro to that. I contacted, like I cold emailed a few labs to see if anyone would be interested in like possibly working on this. And yeah, I've been working with the UCLA's molecular screening shared resource for the past couple of years. That's been pretty cool.
Okay. And one of the challenges in a lot of projects is getting labeled data.
Mhm.
So did this lab then have the labeled data? Like if you had a compound, was there some kind of rating on it if it was effective or not for a particular trial? And I know, I mean, was this related to drug discovery? So there's both efficacy and side effects that
Yeah,
are two
so
things to predict.
yeah, so the data mostly comes in the form of like the compound and its efficacy. I'll go more into that in the presentation, but yeah, the thing about like bioinformatics or like machine learning in bio as a whole is that there's not a lot of data, and a lot of the data
Right.
is not that great. So that's
Yeah.
the biggest challenge of it for sure. Um, but no, I didn't really use data that they had given me. It was more I was working on sort of their projects, and they were kind of advising me on my own.
Okay. I mean, I didn't
Yeah,
know if they had an existing GitHub that had things that they were working on with one approach, and then you were taking a slightly different
oh no,
approach using the
yeah,
same data set that was labeled. So that way you could have a comparison of your architecture. Kind of like, have you ever looked at papers with code
yeah,
where
yeah,
they'll be comparing a data set across several different approaches or papers. And then you can kind of see a chart on the same metric
right,
for that. So can you say your name again?
yeah. Anirudh Venkatraman.
Okay, let's go ahead and get started to tonight's ACM, or Association of Computing Machinery talk, data science talk. So we have a speaker on nanobody optimization via machine learning. It's by Anirudh, and he's at Homestead High School. So this was a science fair project, but it definitely sounds very interesting. A little bit of background about our organization. The San Francisco Bay Area ACM was founded in 1957 to promote the knowledge of modern computing. We want to create a community, support networking and hiring. There's a $20 annual membership. There's upcoming talks on our meetup site. So search with SF Bay ACM on Meetup. And our past talks would be on our YouTube channel, SF Bay ACM org. We try to, but don't always have two monthly meetings, a general computing one and a data science SIG. For the job announcements, if you want to post anything, if you're on the Zoom, there's a chat function. And then if there's a data science related data science related job that your company is hiring for, then you can post a link there. And then maybe you have your name and some contact information. That way somebody can follow up if they have further questions, if you want to share that. Some upcoming ACM events on March 11th and March 12th. We have a 2-day hands-on workshop, scaling computer vision workloads with Ray. The price is $55 to $75. There's a registration there. If you want to take a screenshot or a picture, you sign up through Eventbrite. And then on Wednesday, March 15th, we have human-centered design for VR training. On Monday, March 27th, we have smaller, not bigger data, securing an interconnected world. And then stay in touch on our meetup site for upcoming talks. So I was just informed that we're not going to be judging at the science fair this year. Bill, are you online? Is there anything else you or anybody in the ACM Council wants to comment about this?
Well, the fact that we've been doing this for a number of years, the judging, and there are over 1,000 projects, over 1,000 students in middle and high school, and they come up with amazing projects, some of which are the past ones are listed below. But what we have to do is sort through all these 1,000 and come up with the computer-related projects, judge them, and award cash prizes to the top judged projects. And we hope to go back to judging next year and in the future. And so we welcome people who would like to let us know about their interest in being judges. I see also that there's a note in the chat that on the 29th of March, there's going to be a talk, Improving VR AR Experiences by Understanding the Human Vision System. So that is a joint talk with SIGGRAPH, I believe. So that's all I wanted to say about it.
Okay, very good. So I'll go to the next slide. So we also have a call for volunteers. So you can start with a limited engagement. It doesn't have to be a big effort. Volunteers can join us monthly, and it can be kind of oriented towards the kind of skills you want to use. You know, some people like to do more working with a website. Some people like to work more with sponsors or finding speakers. Or working with a membership. So there's a variety of different types of things that people can do. So that we do have a meeting once a month, a planning meeting, where the board of a dozen of us get together to volunteer to help out planning this. You can, on Zoom, you can use the chat or Q&A to submit questions. So now to introduce the talk. So nanobodies are antibody fragments that mimic humans' natural immune response to diseases. Unlike small molecule drugs, nanobodies rarely have toxic side effects. They're not really recognized by the biological system that much. So they can cross the blood-brain barrier. And they've had promise in treating diseases like Alzheimer's, Parkinson's, and Huntington's. The identification of optimal nanobodies requires expensive and time-consuming lab-based procedures. To address this issue, the algorithms developed in the study computationally predict the nanobody's effectiveness in neutralizing antigen targets and propose a pipeline to create novel, effective nanobodies against antigen targets in vivo, or de novo, I should say. The ensemble in this study is composed of 9 models: an artificial neural net, 5 convolutional neural nets, 3 LSTMs, which make up the numerical predictions of the binding effectiveness for the nanobodies. The predictions are then stacked or used as features for a higher level interpreter model. The predictive model, termed EnsembleStack, predicts the effectiveness of nanobodies with a Pearson correlation of 88 and of 78. A generative algorithm developed by the previous researchers creates the novel nanobody sequences, which are then filtered for their effectiveness via the ensemble stack. The highly effective nanobodies are then used to re-parameterize the generative algorithm. Therefore, multiple generations of re-parameterization, the generative algorithm becomes conditioned to produce more effective nanobodies. So the nanobodies are 6 times more novel when evaluated. Let's see. And I'll switch over to the bio. I will find out more in the talk. So the speaker is a senior at Homestead High School, enjoys music, science, and cooking. The past year, he won 4th place at the International Science and Engineering Fair for his project De Novo Nanobody Design using neural nets, AlphaFold, fold, and docking algorithms, in addition to numerous other awards in state and local levels. He has a passion in the field of bioinformatics and in particular drug discovery, which he hopes to pursue in college. Apart from science fair competitions, he performed cell-based research at the Molecular Screening Shared Resource at UCLA under the guidance of Professor Robert Deminoso. In his free time, He loves to play the drums and cook elaborate dishes with his sister and cousin. So if we were in person, I'd invite applause, but right now I'll stop sharing and we can switch over to the speaker.
Yeah. Hi, everyone. Thank you for the opportunity, and thank you, Greg, for the great introduction. And yeah, I'm really excited to be presenting to everyone today. This is the second time that I'm presenting. The first time I presented, I was going over a similar project in which I had done nanobody optimization, but I'd like to go into detail about it and expand upon new aspects that I think elevate it to the next level with new techniques. So, okay. Yeah, so as Greg had said in the overview, basically nanobodies are therapeutic tools and they're very similar to antibodies. They're different in the fact that only camels and sharks can produce them, and they're much smaller than antibodies, but retain the same amount of binding affinity. And computational nanobody optimization is a relatively unexplored field, and not many people have gone that in depth into it, which is why I decided to try my luck in creating novel nanobodies. So the algorithm that I built presents a full generation and optimization pipeline and improves upon previous methods. So it generates and optimizes the CDR3 sequence of a nanobody. And if you were to think of a nanobody as a protein, which is just a sequence of amino acids, the CDR3 sequence of amino acids is the region of that protein that is most critical to determining how effective the nanobody is. So changing the CDR3 sequence allows the nanobody to be either more effective or less effective in treating a disease. And what the algorithm does is that it optimizes the sequence and predicts the nanobody, the binding affinity or the effectiveness of the nanobody, also termed enrichment, given this sequence of amino acids. So going into some of the related work, basically, researchers at MIT, Jin et al., developed a refined GNN, which is a graph neural network that employs sequence structure co-design. I'll go more into that a little bit later. And Liu et al. Of MIT also developed a neural network, an ensemble using 1 artificial neural network and 5 convolutional neural networks to predict the enrichment or binding effectiveness of a CDR3 sequence for an antibody against the antigen target ranibizumab. So the antigen target is sort of the protein that you're testing your nanobody against. So we want to make sure that our protein has the highest effectiveness against this particular target, which is ranibizumab. So what Louis et al. Sorry.
Just a question. So the audience is going to be more computer science and data science, less about bioinformatics.
Sure.
So can you help us? What's the motivation? Like this disease affects what kind of thing medically? So for people that might not know, and I'll say I certainly don't know what ranibizumab is. What condition is that related to?
Sure. So ranibizumab is actually, it gets kind of confusing. So ranibizumab is actually an antibody in itself. And what you are doing is building a protein that's able to bind to ranibizumab. And this is basically a case study in the fact that if you're able to build a protein that is optimized to this particular target, it should be able to be optimized against any target. So
I see.
Other than ranibizumab, we can choose any other protein that arises from a viral disease, say HIV,
So just
COVID-19.
picking something with a particular shape, seeing if you can fit that shape.
Exactly.
And if you can fit this shape, then you can probably fit other shapes. So it wasn't like anything that's not like this is breast cancer or skin cancer. It's just a early stage of fitting molecules.
Right, exactly. It does still have like some applications. For example, if you want to basically test how much of ranibizumab is in the body, you can do that by optimizing this protein against it. But it's more of a general sort of process, and this one was the target because, first of all, there was a lot of training data available for this particular target. And second of all, I chose it because the previous research has conducted optimization against ranibizumab, and I wanted to test my results against their previous results to compare it. So that's why I chose this target. Yeah.
Makes a lot of sense. Anything that is data rich is a good priority. And I imagine if you wanted to generate more of the antigen, it's just like, if this is the antigen, then you cast something that fits it, then you can find another one where you can kind of simulate the antigen, kind of like casting in metal. You have a negative cast and a positive cast.
Yeah. So you kind of, for a lot of diseases, you already have the antigen target. Like, for example, for COVID, there's the SARS-CoV-2, which is the antigen target that researchers have identified. It's just that there's not that much training data available regarding that. So that's why I had to choose this target instead.
Okay. Thank you very much. So sorry to interrupt, but
no
I'm just trying to represent the audience.
Yeah. So what Rui et al. Had performed was a gradient ascent procedure in which they optimized the highest enriched sequence derived from a previous in vitro experiment. And what the gradient ascent does is it basically guides the neural networks within the ensemble in editing the CDR3 sequences such that the sequence binding affinity is maximized. However, there is no guarantee that the edits that are made to these sequences would produce proteins that are able to be synthesized in a lab, and it only optimizes the sequences from the training data, which therefore comes back to the fact that it's not producing anything novel. It's only sort of editing things that already work.
Could you give a few words about how the gradient ascent does that? That is not something that I think of as being something related to machine learning.
Yeah, so I can go into that a little bit later once I get into the one-hot encoding.
Okay.
I'll go into that in detail a bit more.
Okay.
Yeah, so basically Yang et al. Of Stanford proposed an optimization process called stochastic iterative target augmentation. And what this does is it optimizes actually small molecule drugs against an arbitrary target. But the cool thing about it is that it retains the semantics of a drug structure. So it retains the fact that a drug has to follow these certain properties in order for it to be viable and synthesizable in a lab. And it does that while still optimizing its binding affinity to the target. And what this required was a generation algorithm to generate a library of small molecule drug candidates and a filtering algorithm which predicted the binding affinity of each candidate within the library. However, there is no current in silico technique to optimize the CDR3 sequence of nanobodies against the target and to generate such sequences from scratch, which is why I decided to embark on this project. So going into like a visualization of it, the framework that I used was a humanized nanobody framework. So as you can see, this is the framework on the right, and this is the framework that Liu et al. Used for targeting ranibizumab on the left. So as you can see, the framework I use is much smaller and also has a lower polyreactivity. And in order to, so polyreactivity basically means that it is not as prone to giving false positive results. So a lot of these nanobodies and antibodies, when you test them in a lab against the protein you're targeting, they would sort of bind to anything. And that would tell you that it is, although it is binding, it is not binding very well. And it will just bind to any single target in your body, which is not good because that will produce a lot of different side effects. So you want something with a low polyreactivity or high specificity. And that's why I chose this framework. And as you can see highlighted in blue, that little region, that is the region that determines the binding affinity of the nanobody. And that's the CDR3 region in blue.
So are molecules actually getting produced based on what you're doing and then tested in the lab? Or are you simulating this? Or how do you calculate a metric for polyreactivity?
Yeah, so the polyreactivity comes from the fact, it comes from the previous paper, which was published about the nanobody framework.
Okay.
Yeah, and yeah, it has a lot of details there. I can link the paper in the chat. But I
But
also
just in a summary, there's code you could get from GitHub from a paper that could calculate the metric that you're trying to optimize on. In
yeah,
this case,
exactly.
the polyreactivity.
Yeah.
Okay.
Yeah. So quickly going to the method overview. So what you have is your generation process, which generates a lot of different candidates for CDR3 sequences. And as you can see, these are just about 13 to 20 letter sequences of amino acids. And this is actually represented in the FASTA format, where each amino acid is designated by a single letter. So as you can see, A is alanine, T is tyrosine, G and M are the respective amino acids. This passes then through a binding affinity filter. So you have this other algorithm that I created called EnsembleStack, which I'll go through later, in which it takes these candidates of the CDR3 sequence and is able to predict how well they would perform as part of the Nanobody framework and test it against the antigen. And if the binding affinity is high, it passes through this filter and is used to readjust the parameters of the generation algorithm. So over multiple generations, you're able to see that this generation algorithm gets re-parameterized such that candidates over time become higher and higher affinity. And this kind of converges to candidates that are much more similar to each other. So yeah, I'll go through everything in detail a little bit more. So one thing that I had to make sure was important was that the generation algorithm was as sound as possible, because otherwise you could be just generating a bunch of noise. And a lot of times the nanobodies that you generate, if you keep re-parameterizing the generation algorithm, it might converge to random noise or just random data that's able to sort of cheat the filtering algorithm. And what I wanted to do was make sure that the CDR3 sequences that were generated were once grafted into the nanobody, this would create a biologically sound structure. So in order to do that, I leveraged Jin et al.'s algorithm, refined GNN, in which he used a graph neural network to train a machine learning he trained a graph neural network to predict the sequence and structure of a CDR3 sequence in the context of an entire nanobody. So what it does is it sort of incorporates information about three-dimensional structure, and these include the dihedral angles and distances between alpha carbons. And the dihedral angles are the angles between the bonds, and the distances between the carbons in the amino acids are also incorporated. And this creates CDR3 sequences with a greater understanding of the actual biological context. So as an example, yeah, sorry.
So were you able to prune things that were not physically plausible? I mean, because you can generate a sequence of letters, but then once you calculate the angles, if two things overlap in 3D space, it's not plausible. So there's a way of calculating what's physically plausible and not plausible.
Yeah, so this already has like that filter through it. And
Okay.
it calculates which is plausible, which is not plausible. Like if it overlaps in 3D space, like obviously that's sort of an error we can catch in computationally. But the thing about this graph neural network is that it's more likely to produce sequences that are plausible in nature. And this has been verified through its through testing data, and it's been shown that it is able to produce actually better, more higher quality predictions than AlphaFold.
So it's kind of like in linear programming or nonlinear programming, you have constraints. Don't search certain areas that are not plausible. Your optimization. Go ahead, Bill.
Were there particular libraries that you used to implement the neural networks, the graph neural networks?
Yeah, so PyTorch usually kind of just worked for everything. It was more of like a one-size-fits-all for all the neural networks used here. This was already implemented in PyTorch. So I decided to implement the rest of the algorithm as well in PyTorch.
Okay, I mean, was there already a body of routines that were implemented in PyTorch that that were kind of a higher level than just PyTorch itself that you were able to use?
Um, Actually, not really. It was mostly just PyTorch itself. I think there was a little bit of using PyTorch 3D, which is just a library on top of PyTorch that implements sort of all the graph convolutions in a more simplistic way. But yeah, I think at the highest level was PyTorch.
And does that mean that you have created some code that is now available for other people or
Yeah,
could be used?
yeah. So this is all on GitHub. I can post the GitHub link in chat as well. Then if possible.
Yeah, that would be
Okay, if
interesting.
you're comfortable.
An audience question on chat. Michael
Mm-hmm.
is asking, what is CDR3?
Yeah, so the CDR3 region is the region of amino acids within the protein that determines its binding affinity. So it is the most important region within the protein for determining how well that protein would bind to another target.
Kind of the active region where things are happening.
Yeah, exactly. The most active region of the protein. The protein or the nanobody. Nanobodies are proteins. So yeah, just a quick example of how the graph neural network works. So the features that were used were, all the node features that were used were the dihedral angles. So this is the angles between the carbons and the nitrogens, the carbons and the oxygens, and the carbons, alpha carbons and the beta carbons. So these just govern sort of the orientations of each atom. The edge features include the distances between the atoms and a few orientation features to capture in what state the protein is in, because proteins are very, they're not that rigid. So basically they could fold in many different ways. So implementing these orientation features kind of allows for for a method to take a snapshot of what position the protein is currently in. So using these node and edge features, we're able to sort of predict what the next amino acid of the protein would be and sort of the position of that amino acid. And by iterating over this algorithm, you're able to sort of compute the loss between not only the structure or sort of the angles and the distances, but also the cross entropy loss of what the predicted amino acid type would be. So at the same time, you're able to predict a sequence, and you're also able to predict its structure. So switching gears a little, basically, once you have a sequence that has been generated by the graph neural network, what I do is I one-hot encode the sequence. So CDR3 sequences can range anywhere from 13 to 20 amino acids. So in order to make all this data uniform, what I did was I buffered these sequences with the letter J at each end until it was 20 characters long. And then I one-hot encoded it. So basically you have, I'm sure most of you know what one-hot encoding is, but to go over it, you have the mapper, which maps each amino acid type to a particular, it just maps each particular amino acid type to a particular integer. And you're able to represent a sequence as a matrix, and you're able to basically iterate upon this matrix with the neural network. So as you can see, sort of with the mapper, when the I matches up with the I, it produces a 1, and sort of over the entire sequence, you're able to get you're able to get a 20 by 20 matrix, because there are 20 amino acid types and 20 characters in each CDR3 sequence. Yeah, so in order to predict the enrichment or the binding affinity of the CDR3 sequence, what EnsembleStack does is it performs a weighted average of the enrichment predictions from 9 more naive neural networks with different architectures. So you have artificial neural networks, convolutional neural networks, and a long short-term memory recurrent neural network, or LSTM-RNN. And this process is known as stacking. So With each of the 9 neural networks, you train them on 60,000 different CDR3 sequences and their corresponding binding affinity against ranibizumab. So each of these sequences are represented as its one hot encoding, and they're passed through each of these 9 neural networks for prediction of its binding affinity. And once you get that numerical binding affinity prediction, you average it or perform a weighted average using an interpreter neural network to determine the final binding affinity of that particular sequence. So yeah, kind of going through each of the different models. So you have each of these 9 different models here, and their parameters. Yeah.
Question on chat. How are the 20 amino acids mapped into the matrix? So I assume each amino acid was a letter.
Yes. So you have the mapper, which has each amino acid as a particular letter, and you have your sequence over on the left-hand side, and they're kind of mapped to whichever one represents it in its mapping space.
So like in the middle there's 2 S's in a row in the sequence where there's 2 adjacent ones.
Yeah.
So the sequence is the order of amino acids, and then the mapper is on the horizontal axis is which of the 20 amino acids. Is that the case?
Yeah, that's the case.
Okay.
Yeah.
And then on your most recent slide that you were speaking to, how do you know the ground truth? A neural net's trying to predict something. It's coming up with an estimate. You calculate an error between an estimate and an actual. So how do you know for new molecules what the ground truth is?
Yeah. So in the training data, you have 60,000 different CDR3 sequences and their binding affinity for ranibizumab. And this was determined in a lab. And this was performed by researchers at MIT. And you're using this to train 9 different neural networks to predict the binding affinity of a random CDR3 sequence.
Okay.
Yeah.
And then my own, going back to that slide, my own observation, as opposed to just a weighted average of a number of neural nets, something that you could consider as a future work is train a regression or an XGBoost just to create your own weights. And especially something like an XGBoost can be, have very local kinds of interpretations. So given this context, might have a different weighting, whereas a regression might have an equal weighting on the different neural nets. So like some of the substructures, like LSTM number 2, might be very good in certain angles and certain molecules. Whereas LSTM4 might be much better in others. So as opposed to just having a blind equal average for everything for all molecules.
Yeah, so that's actually what I did. Instead of an equal average, I was going to go into that. I created an interpreter network, which is able to take all these predictions, and it has a hidden layer within it and come out with a final prediction of the enrichment.
Okay.
Yeah. So going into the different model types, you have the ANN, you have only one, which is around 13,000 parameters, and they all have relatively small amount of parameters. The highest with 46,000 it's a decent amount, but it's not too much to overfit. And you really don't want any of these models to overfit the data, because you want more naive predictions in order to sort of get a general idea of how the regression works, in order to, once we combine them together, get a much better idea of the binding affinity. I think we have a few questions in chat as well. Okay, all right. Yeah, so moving on to the interpreter neural network. So each model of these 9 models, they're trained on 3 different data sets, each data set consists of 60,000 different CDR3 sequences. And over 3 data sets, there are about 100,000, with some of them overlapping. So once you pass the CDR3 sequence through the model, you're able to get 3 different predictions of its binding avidity or its enrichment. And each of these 3 different predictions are used as features for a higher level interpreter neural network. So these are the parameters, and it's get passed through a 16-neuron dense layer for a final prediction of the enrichment. So it takes all these 27 different parameters and then passes them through a hidden layer of 16 neurons, gets a final enrichment. And this enrichment prediction is actually much better than each of these constituent predictions of binding affinity. Yeah, so moving on to the optimization process. So the EnsembleStack algorithm filters the initial generated library of 20,000 sequences. And this was going back to the graph neural network. This was generated by the graph neural network, all these 20,000 CDR3 sequences. All of these sequences are passed through the algorithm, and candidates with an enrichment less than 0.5 are thrown away. And the higher affinity candidates with an enrichment greater than 0.5 are used as training data to re-parameterize the graph neural network. And this is known as stochastic iterative target augmentation. So going into sort of the re-parameterization process, I can go back a couple of slides. Sorry. So as you can see, like you have each of these amino acid types that were used as part of the generation algorithm. And in order to re-parameterize the generation algorithm, you take the sequences with high affinity and you compute the cross-entropy loss of the amino acid types with the sequence generated and the sequence with the high affinity. And you back propagate on that loss in order to create higher and higher affinity candidates over time. So you're not training on any structural data at all. You're only training on sequential data. But the good thing about it is that the graph neural network already retains its structural parameters from its previous training when it trains on the structural components of antibodies and nanobodies with the node features of the dihedral angles and the edge features of the distance and orientation features.
So my ignorance in bioinformatics, so everything is a linear sequence. There's no rings, there's no forks, there's no dags, there's no trees. It's just always a straight line.
Yeah, it's always just a linear sequence. So,
okay. That does simplify some things.
Yeah.
I think one of the questions that was asked about how did you split the CR3 data for training and testing? Let's see. You on your slide, you talk about how it's used to train the next step, but what about the first step there? How did you decide? Did you just do like this percent for training and this percent for testing, or did you do something else?
Yeah, so this training data was already previously used for Lou et al.'s experiment, where he didn't use sort of the stacking method. He only used an ensemble. And the thing is that I copied the same train test split in order to maintain the accuracy and make sure that it's kind of a one-to-one comparison of my algorithm versus the previous ones. And as for training the interpreter neural network, I used about 5,000 sequences from the testing data. So.
Okay, and you just now mentioned that that previous work did not use some of the stacking, I think is what you said.
Mm-hmm.
So that is one of the things that you added with your work.
Mm-hmm. Exactly.
And so that'd be interesting as you continue to talk to point out, the additional features that you or the differences that you got into beyond what you were building on.
Yeah. So stacking was the main difference that differentiated my algorithm from Lou et al.'s. But another main difference is that kind of my algorithm includes an entire complete process of generating a nanobody from scratch rather than iterating upon training data. Because the best nanobody sequence may come from something that hasn't been seen before in the training data, which is why I included an entire pipeline.
A question in the Q&A, Carl was asking, how does the final enrichment prediction get computed from the neuron dense layer?
Yeah, so it's basically a simple regression model. It's just a 16 neuron dense layer. It computes sort of a weighted average of it using its regression parameters and weights in order to come up with a final enrichment prediction. And it is trained so that it uses the mean squared error loss function in order to get as close as possible to the ground truth values. And that's how the regression model is trained. It just adjusts the weights of the dense layer accordingly.
No special transfer function.
No.
Sure.
Yeah, so going back to the optimization process, the high affinity candidates are used as training data to re-parameterize the graph neural network. So the sequences that are generated by the graph neural network, you backpropagate on the cross-entropy loss function between that sequence and a ground truth high affinity sequence. And you use that to sort of have the graph neural network generate higher affinity candidates over time. And the thing about this is that although you're only training on sequential data, the graph neural network is able to retain its same structural parameters, as I said previously. Yeah, it minimizes the sequence-wise difference between the true and predicted sequence. So this is just sort of a pipeline of how the optimization algorithm works. I can go through it a little bit. You have the generative graph neural network G, the enrichment predictor E, and the nanobody context region C. And the nanobody context region is just the regions of the nanobody excluding the CDR3 sequence. So in each generation, you generate a CDR3 sequence using the graph neural network and append it to a data set. And for each sequence in the data set, you predict its binding affinity or its enrichment. If the enrichment is greater than 0.5, you append the sequence to a filter data set Y. And for one epoch, you train this graph neural network, the initial one, on Y to create a new iteration of the graph neural network. And you do this I did this for 5 generations. And by the end of the 5th generation, I was able to sort of converge to sequences that look very similar to each other. Yeah, so going into some of the training data analysis, what I wanted to sort of show was the ability for neural networks to parse these biological data. Basically, I built a TSNE plot of the the CDR3 sequences for the training data. I'm sorry. And basically what you can see from these graphs is that sequences with a high enrichment or a high binding affinity are very close together, which indicate that they have these patterns that are able to be picked up by a neural network compared to sequences that have a low binding affinity. This just indicates that for the antigen target ranibizumab, there is a specific pattern that most of these CDR3 sequences have to follow in order to be a high affinity sequence. And this also goes, if you look at the second graph, this is for sequences with already a high enrichment, an enrichment greater than 0. And the really, really good ones also tend to follow very similar patterns. So there is a pattern that is able to be determined by a neural network in order to find sequences with a high affinity. And that's sort of just in itself showing that this project is sort of viable, just as a foolproof way to figure it out.
The blue clouds on the right of your previous slide are showing there's subspaces that are interesting and very useful, that are very local and not global. And it's not kind of randomly distributed. It's kind of in clusters there. So.
Yeah, exactly. And the thing about it is that this is for sequences that are already have an enrichment greater than 0, which means that it's already going to bind. So among these sequences, the ones that are high binders or the darker blues tend to cluster together as well.
Right.
Yeah.
And then is there something you could do with that to almost do a clustering where you see things above a certain, darkness of the blue enrichment level, and then just kind of continue searching in those areas? It's almost like a genetic algorithm. Organisms that were previously successful, you kind of search in that subspace more.
Yeah, that's a really good idea, actually. I haven't done that yet, but it'd be pretty cool to see, like, at what point do these sequences stop clustering, and where does it sort of taper off, where
and then also
binding enrichment.
The clusters can give you a good idea for originality, because if you have some known compounds in some clusters, that's kind of like a cluster of known stuff.
Yeah.
If you find some new clusters that don't have known compounds, then that's a good way of demonstrating, hey, I found something that's more original.
Yeah, exactly.
A new search space so when they do the strategies for physical drug discovery, they can kind of take the clusters into account for what they might want to do for their design of experiment strategy.
For sure.
A comment in Q&A from Kim. She said, "This work is very impressive in my view, way beyond the scope of most Homestead seniors. Both my son and daughter graduated there. At the end, I'm interested in how he got up to speed in this very high level of technology. Mentors, reading papers and publications. So how did you get up to speed in this?"
Yeah, so I've been sort of researching this area for about a year and a half, maybe 2 years. So it's mostly just kind of reading papers and kind of doing these projects on my own. A lot of it was just like reading papers. I didn't really have too many mentors apart from my like occasional meetings with my professor at the molecular screening shared resource, but most of it was just, I would say, reading papers and reading blog posts online. There was a lot of information available on bioinformatics in particular, and they really break it down to very simple levels.
Okay, yeah, that's what I was going to say, is because also you're going to have kind of get the basic terminology or jargon of that are journals before you can kind of get very far in the journals.
Yeah.
On the chat, somebody said, "What are the full dimensions of the T-SNE components?"
So they're 20 by 20 matrices.
Okay.
Yeah. Each dot is a 100 coded matrix, which is 20 by 20 dimensions wide.
Okay. Thank you. Oh, and then Kim responds on the Q&A. She said, "Thanks, great job." So
thank you.
Just to pass on the positive feedback.
Thank you so much. Yeah, so going into some of the results, this is just a regression graph of the predicted enrichment versus the ground truth enrichment. So as you can see, it follows a relatively linear curve. The R squared is around 0.78, and the Pearson correlation is 0.88. And one interesting thing that you might notice from this graph is that the sequences that have a predicted enrichment of greater than 0 are much closer together than sequences with an enrichment that are less than 0. And this kind of goes to show that it is it's like a no-nonsense test, that it is easier to create a sequence that has a high binding affinity because it follows a pattern rather than make a bad sequence, which can basically fall into any bucket or any pattern. So that's why it's really bad at predicting exactly how good bad sequences will be, but it's good at telling how good sequences would be. And it also shows that this algorithm is implicitly a good classifier as well, despite the fact that it is trained on regression.
So, Anirudh, I don't know if this comment that I'm about to make makes much sense, but I do notice that there's a really strong barrier there, or boundary in that graph. And is that kind of like an artifact of the numbers that are coming out, or is there something meaningful going on there?
Yeah, so this boundary comes from the fact that it is like implicitly classifying between good and bad sequences, and good and bad sequences fall into either if it's positive, that means it is a good sequence. If it's negative, it is a bad sequence. So it's sort of able to tell which ones are good and which ones are bad.
Actually, I'm talking about the boundary. I guess it's not quite a 45-degree angle that thing goes up at, but kind of the bottom that separates it between the upper left and the lower right. Very strong boundary there.
Like right here, can you see my cursor?
Starting at the lower left and going up towards the upper right, you've got a really clear line there. And I was just wondering if there was any real world significance to that, or is it just some kind of artifact of the math?
Yeah, so I think the real world significance of that is the fact that there's not a lot of would it be false positive? Yeah, false positives, because it barely predicts any sequences with a ground truth enrichment of less than 0 to be very high. So I think that's the real world significance that comes from that.
Okay, thanks.
Yeah. Yeah, so going further into the EnsembleStack results, basically you have here in the box plots are the results from the single models, and the blue dot is the result from the ensemble interpreter. So the constituent neural networks, because they're very naive, they produce a lot lower accuracy estimates than the stacked interpreter. So you can see there's a drastic difference in mean squared error, a Pearson correlation coefficient, and in R squared as well.
Excuse me.
Comparing. Yeah.
Where is the blue dot that you're talking about?
In the stacked interpreter, it's the yeah.
So it's a box and whisker plot where the confidence interval is so narrow, the box is reduced down to a line.
Yeah.
So it's showing very consistent results, which is very good.
Yeah.
If it was a wider box and whisker column, then they would have higher noise or variation.
Yeah. So each of the red dots in these box and whisker plots are actually each of the different types of neural networks. So you have the 3 artificial neural networks. You have the artificial neural networks. You have the 5 convolutional neural networks, and you have the 3 LSTM-RNNs. And these kind of constitute the red dots in each of the box and whisker plots on the left. I think there's a couple of questions. Yeah, so the scatter on the left is much higher than the scatter on the right. Yeah, so this sort of indicates the fact that the ensemble is not that great at predicting the exact binding affinity of the lower enrichment CDR3 sequences, because it is sort of unable to tell how bad a sequence is, but it's able to tell how good a sequence is, because there's a lot of ways to go wrong in constructing a CDR3 sequence that would not have a high binding affinity against the antigen.
Is that also related to when you had a cutoff at half when you were adding to the training data? So you're just training on the more effective bindings. You weren't training as much on the less effective bindings. So by having less training on the less effective bindings, the model's not as good in the less effective bindings. Is there a relationship there, or?
There was actually an equal distribution of the positive and negative bindings. So yeah. Yeah, so, but an also indication of this is that the algorithm has a pretty it has a relatively high false negative rate. So that's one thing to be improved upon in this algorithm. From this section right here is the false negatives.
Could you improve on that with just data augmentation? You
Yeah.
know, records that you're weaker on, just duplicate them 10 times in the training set, disperse them randomly.
Yeah, I think data augmentation would definitely be helpful, but probably the biggest thing is just getting more data. And that's the thing with most bioinformatic experiments, that there's not much data available. Yeah, so moving on to the results. Basically, I compared the results of my model to the non-stacked model that was created by Liu et al. And what I noticed was that my mean squared error, Pearson R, and R squared were all more accurate. And this is sort of case in point that the interpreter neural network is able to average each of the constituent neural networks in such a way that predicts the most accurate estimate of binding affinity compared to the previous methods, which was just a straight up average and a voting system. So yeah, just to show it is more accurate on all three metrics. It's all tested on the same testing data as well. Yeah, so moving on to the optimization results. Basically, this graph right here just kind of shows the enrichment or the binding affinity of the binding affinity distribution of the nanobodies over multiple generations. So as you can see, over each generation, the enrichment, its tail gets longer and it's approaching a more normal distribution. And the tail getting longer indicates that over multiple generations, it's producing more highly enriched CDR3 sequences, which is an indication of the fact that this optimization pipeline is working. And just another cool thing that I saw was sort of the amino acid compositions of these sequences compared to the original training data that was used to train the ensemble. So the optimized sequences have a much different sort of composition than the original training data, which is interesting because it shows that these sequences are novel. But it was also a little worrying to see that it doesn't totally follow the same pattern. But I'll get into how I confirm that later through an in vitro experiment. Yeah, so what I actually did was I took one of the CDR3 sequences that I had optimized from my experiment, from the optimization pipeline. So it had a much higher predicted enrichment, and its perplexity, or the sort of confidence in its biological viability from the graph neural network was very low, which indicates that it would perform, it would be a biologically viable sequence, more likely. So what I did was I performed an ELISA test, which is a test to see how strongly it would bind to the antigen target. So I took an antibody, which was specifically designed to target ranibizumab, and I used that as the positive control. And I took my nanobody and also tested it, and the last two wells were the negative control. And a higher sort of fluorescence from each of these wells indicates a stronger binding. So as you can see, the nanobody does not indicate as strong of a binding as the positive controls. And that just indicates that, well, the positive control was sort of specifically designed to target ranibizumab, and it was specifically tested in a lab. So I was kind of thinking that there was a very low chance it performed that much better than it, but it still indicated binding, which was a nice confirmation of this entire experiment. And the nice thing about it, sorry, is someone speaking?
Yes, I just wanted to clarify for myself here. Does this mean that you actually created, somehow got a hold of the actual biological implementation of these protein sequences, these antibody, what do you call them, amino acid sequences?
Yeah, so I took the amino acid sequence for the CDR3 region, and I cut and paste that into the Nanobody framework. And I was able to contact a company which was able to synthesize this nanobody. And yeah, I used that as a biological test against the antigen target. So it was all sort of synthesized and tested in vitro. Yeah,
Oh,
so
great. Yeah.
ELISA stands for enzyme-linked immunosorbent assay. And it's just a test to see how strong an antibody would bind to its antigen. And the stronger it binds, the more fluorescent, or the stronger the color is. And the less it binds, the less the color is. But the color in general indicates that it is binding to the target. So it was nice to see that the CDR3 region that I generated and grafted into the Nanobody framework was actually able to produce an antibody that is able to bind to the target. So this is just a quantification of the ELISA results. Absorbance is the same thing as sort of the fluorescence. The positive control exhibits a nice smooth curve, although the nanobody is a decent amount worse, but it still indicates level of binding, given the fact that it produced a level of absorbance that is necessary to indicate binding. Yeah, so kind of synthesizing the whole thing, this entire project developed a full computational pipeline for both generating and optimizing nanobodies from scratch, creates novel nanobodies. It makes more accurate predictions of enrichment than previous ensemble methods using this interpreter neural network. And it created an antibody CDR3 region from scratch, which indicated binding when tested in the lab. And here are my references. So, yeah.
How do you evaluate novelty? So you were saying can generate more novel things.
Mm-hmm.
There was an earlier slide where you had blue clusters of dots. And so if there was a lot of known compounds in a blue cluster, that might be, and you discover something that might be less novel, finding something in a new blue cluster might be more novel. Is there some novel calculated KPI from one of the papers that was accepted?
Yeah, so novelty, it can be tested through Hamming distance as well.
Okay.
Yeah, and I think what I used was Levenshtein distance to indicate novelty. I don't think I included this graph in here, but Levenshtein distances indicate just how many mutations are necessary to convert one sequence to another. And the Levenshtein distance
just for the general audience, that's used in spell checking for like an edit distance. Like if you have a misspelled word, then transposing two letters, or how many edits do you have to do to match a misspelled word with a correct word in a dictionary. So he was using that in his, go ahead, continue, just to give background for the audience.
Yeah, no worries. Yeah, so for the previous experiment where they used gradient ascent to generate new nanobodies or new CDR3 sequences, the Levenshtein distance was around like an average of 2 to 3, but the Levenshtein distance from my sequence that I generated from the training data was around 13. So that just indicates like it's much more novel compared to the training data. And these sequences are around 13 to 20 characters long. So it's almost sort of a entire revamp of the sequence itself.
So that'll be like 13 edits out of a 13 long string, or a 28 long character string. Yeah, so that is very, very different.
Yeah.
Other questions?
See, Carl has something about a 100% holdout per test. I'm not sure what he means by that. Is that, or no, 10% holdout per test? Oh, okay, so he's asking again about the idea of how did you create the test versus the training split, I think is
yes.
what's getting at there.
So yeah, so going back to that, the train test split was the same that was used in the previous paper in order to get a one-to-one comparison of my interpreter network in predicting the binding affinity versus the previous algorithms ensemble method of predicting binding affinity. So it was around with 60,000 CR3 sequences. I think the testing data was around 20,000 sequences.
So in this testing now, so you've done a bunch of this predictive work, and the previous researchers had done a bunch of this predictive work. Just out of interest, has there been any implementation in some kind of testable drug for this kind of thing?
So yeah, it's pretty hard right now to do that because like the I mean, FDA approval does take a really long time, but
yeah,
I think they did just release, from what I saw recently, their first AI generated drug, and it has been FDA approved. But a lot of these drugs are showing pretty high promise in phase 1, phase 2, phase 3 trials, which is nice to see.
okay.
What are continued directions that you want to investigate in or work on?
Yeah, so I guess some things I want to work on especially are more going into the in order to improve the accuracy, you can't really get too much from a sequence itself. Instead, what you could do is sort of go into the nitty-gritty of the protein structure and be able to predict that better. So that's what I'm kind of doing, is finding methods to predict the protein structure more accurately in order to get a better estimate of its binding affinity, because the sequence can only go so far. But from
kind of a better physical model.
Yeah, exactly.
Well, isn't the physical model embodied in the angles that you were talking about earlier between different atoms?
Yeah, it is. That's sort of only for the generation algorithm, though. And what I want to do is more integrate that the physical model in the predictive algorithm. So using the physical model, be able to, when I use the physical model, I'll obviously be able to have more features, and being able to do that would be able to get a better estimate of the binding affinity.
And as I say that, I'm the physical modeling that you did have with the angles and so on, was that were those 3D or just 2D type angles?
Those were 3D, so they included like a lot of different orientation features as well, in order to make sure, because the thing about like protein binding is that there's a lot of rotating that occurs. So you need to make an equivariant model that's able to be represented in multiple different orientations. So that's why a lot of different mathematical sort of steps that were a little bit over my head had to be taken by the MIT researchers.
Okay. And again, just something maybe out of left field here, but I remember from years ago, there was this distributed project called Folding at Home, which was about protein folding. And I'm wondering if there's any interplay between what you were doing and that project or projects like it.
Yeah, so a lot of the protein folding algorithms that are like more universal, like Folding at Home, like AlphaFold, like OpenFold, is that especially for antibody binding regions, it's not that great at predicting the structure, because antibody binding regions are so variable and they can be sort of twisted in so many different ways that it's not that great at predicting what its structure should be. So that's why there's been a lot of work recently on using sort of different approaches to predicting the protein structure.
A couple of questions on the Q&A. Carl asks, if you could feedback quantitative ELISA into your system, that iteration could knock out some novel or unexplored regions that were not as productive as expected.
Yeah, so the thing about that is that basically you have to be able to synthesize the nanobody first, which does take a lot of time, and it is pretty expensive. So you kind of need a lot of data in order to get the ELISA result back, and then feedback it into the system, which would require, it's not complicated in a computer science perspective, but in like a logistics, because there's so many different nanobodies that need to be synthesized. It'll cost a lot of money. It'll take a long time. So that's just the sort of logistical aspect of that.
Okay. And then
Yeah.
another thing on the Q&A, SS says, if it's a 3D, I assume meaning the molecule, then do you need two angles to represent it? Is that correct? Or is the angle just a local angle and you just take the smallest angle between two bonds or something?
Yeah, so for each amino acid, it's kind of tough to see. So each amino acid is composed of what differentiates amino acids is the side chain region. Basically, amino acids are composed of usually the same like 4 molecules: carbon, nitrogen, and oxygen. And what differentiates the amino acid is the side chain that's attached to one of the carbons. And the angles that you use are the angles between these carbons and the nitrogens and the oxygens. So you have 3 different angles. The psi is between, I have to recall it a little bit, but the sort of the psi, phi, and omega angles are between each of the 3 carbon, nitrogen, and oxygen atoms. And these sort of represent the orientation of the bonds in space.
So these are but standard metrics used in the literature.
Yeah.
That are standard ways that the chemists think about things, I assume.
Yeah. Yeah. So this is used by AlphaFold, is used by all the protein folding algorithms. It's a very common way to represent the protein structure.
Okay, just the significant features. SS says thanks. Any other questions from anybody or comments?
Looks like there's another question that came in. Is it in the Q&A? Oh, no, it's just a thanks. Okay.
SS says, very nice talk, especially for a high school project. Keep up the good work.
Thanks so much. Yeah.
And Carl says, great work. I'll say the same thing. Looks like very good work.
Thank you.
Okay, I guess if no one has any other questions, I guess we'll say thanks again.
Of course.
And
yeah.
Thanks for presenting again to us.
Let me quickly drop the GitHub link in the chat just in case anyone wants to take a look.
Yeah,
sure.
Please.
Kim has asked, have you chosen a college yet?
Not as of yet. I'm still waiting for the regular decisions to come out later this March. So we'll see.
Okay. Well, good luck.
Thank you. Yeah.
Okay.
Was that okay?
Sounds good. I'll add that GitHub link into the meetup. So if people look at the meetup at the bottom, there's a discussion session. They can see that link.
Sure. Awesome. Yeah.
Okay. Can you post your slides if you can save them as a PDF and then just put a link at the bottom of the meetup as well?
I don't think I'm able to edit the meetup, but
well,
on the bottom of the meetup, there's a discussion session where anybody can post comments or links. And that's where I was going to post a link to your GitHub.
Okay.
So you just go to the meetup for this event and then scroll down to the bottom. And then there'll be a section. Or if you have questions, reach out to any of us at ACM. And we could do it for you as well. Okay. Well, thank you very much. And good luck with everything.
Thank you. Yeah. Thank you guys so much for organizing this.
Sure. Okay. Thanks. Bye.
Okay. I'm going to end the meeting now. Thank everybody for coming. Thanks.